Okay. All right. I view the world as a protein engineer, and my goal is to make tools that people will actually use. And that requires getting a lot of details correct. So I'm not going to focus on any of those details, but rather I'll give you a summary of how genetically encoded fluorescent sensors for neuroscience are designed, and then I'll show you some of the highlights of what they can do. On the last slide, I'll show you how the GABA sensor might be relevant to probing GAP1 function, and potential therapies. Which is the, okay. So, first of all, first question, show of hands, how many people have heard of GCAMP, the calcium sensor? Okay, we got probably a little bit more than half. All right, we'll go through this quickly. Some proteins undergo global conformational changes upon ligand binding. Calmodulin goes from an extended compact shape to a condensed shape when it binds calcium and its cognate peptides. This conformational change can be exploited to make a genetically encoded fluorescent sensor by making a single polypeptide chain containing both calmodulin and GFP and hooking them together in just the right way so that the global conformational change upon calcium binding is coupled into a change in the local environment of the fluorophore. That in turn can change the fluorescence of the GFP and make it get brighter. This calcium sensor is called GCAMP, which is like GFP, but with calmodulin in the middle. Okay, so here you can see an example of the local conformational change in the unbound to calcium bound state. You can see that this arginine goes from being away to actually making contacts with the chromophore. So now GCAMP has been worked on by many people at different institutions for over 15 years. At Janelia, a team was assembled to exclusively work on improving it to the point where it could report neuronal spiking in multiple preparations. In this experiment, virus expressing GCAMP was injected in the mouse visual cortex, and while the mouse was looking at drifting gratings of black and white stripes, the activity of individual neurons was observed through a cranial window. Which neurons light up depends on the directionality of the movement of the gratings. So for example, you can see here the neurons in turquoise only respond when the drifting grating is going up and down. And different variants have been made, including some that can resolve individual spikes. So the calcium sensor is very sensitive and can even detect, oops, sorry. Can even detect, let me go back one. All right, can you restart that, please? Yeah, that's a good enough spot there. Right where you were. Just start anywhere. Start anywhere, that's fine. All right, so key thing here is to show you the movie, which is it can even resolve individual spikes on individual spines. Okay. All right, so now to make sensors like GCAMP but for other stuff, all we need to do is find a protein that binds our analyte of interest and undergoes a conformational change. The periplasmic binding proteins of bacteria is a diverse family that provides many such scaffolds. They all undergo a Venus flytrap-like motion upon ligand binding. Here you can see the stereotypical change of Maltose binding protein. We can identify regions that undergo local conformational changes either by doing some calculations of how the backbone shifts or by just eyeballing it. And you can see here this one little spot has a good torque that exists. And oops, I keep bashing that. And if we insert circularly permuted GFP there, we could potentially get a sensor for our analyte of interest. And that's just what we did with the glutamate binding protein to make an intensity-based glutamate sensing fluorescence reporter, or iGluSnFR. You have to have a cute name if you're going to come up with a sensor. So there's only a structure of the glutamate binding protein in the closed state, so it's by homology to the Maltose binding protein that we decided to put the GFP there. Okay, so there's in a longer talk, I tell you a lot more about the details of protein engineering, but we're just going to skip that for now. And so we made a sensor that works great as a soluble protein when it's expressed in bacteria, but as most neuroscientists would care not about soluble glutamate, but rather they want to know how much is released at the synapse. So we added a secretion signal and tagged the sensor with a transmembrane domain. So it would go to the plasma membrane, be facing the outside, and then when you add glutamate, the cell membrane gets brighter. Here you can just see it in HEC cells gets significantly brighter. You can see here there's some sensor that's trapped in the endoplasmic reticulum, and in the delta F over F change in fluorescence map, you can see that that does not respond. So we were lucky that the affinity was just perfect for detecting glutamate release in neurons, and you can see what happens when we have cultured neurons expressing iGluSnFR, and we give a single electrical pulse. All gets brighter. So in the final figure of our paper describing iGluSnFR, we show blips of glutamate. Oh, no one's going to be able to see this from where you are. All right. Well, you'll have to trust me. There are little dots here that you can see. We showed that there are little blips of glutamate activity in the motor cortex that lit up when the mouse was specifically running either in the forwards or the reverse direction. Now, we have since made a bunch of variants that are more stable, that bind glutamate more tightly with a slower off rate, or more weakly with a faster off rate. And because GFP has a lot of chromatic variants available, we were able to make, modularly make mutations to the sensor. And now we have a palette of iGluSnFR variants that allows different types of experiments to be performed. We can talk about all that later if you wish, but let's get to the matter at hand here, which is GABA. So, while glutamate is excitatory, as we all know, GABA is the primary inhibitory neurotransmitter, except when it's not. They are structurally quite similar. Okay, the only difference is the absence of this carboxylate group on GABA. So one could imagine that you could make a GABA sensor by redesigning iGluSnFR. We tried that, but failed. I could talk to you a long time about the structural reasons for that. One could also imagine making a GABA sensor from the GABA B receptor, which also has this Venus flytrap-like fold. However, it's an obligate heterodimer. It's difficult to express heterologously. Furthermore, there's a significant potential for adverse interactions with endogenous proteins and receptors, and all the pharmacological tools that we have available for probing mammalian neurons would thus interfere with the sensor as well. So we chose not to do that, but rather we turned to another GABA binding protein that we isolated from Pseudomonas bacteria. We inserted GFP, and we optimized it like we did for iGluSnFR. We got a great in vitro sensor. You can see it here in black. But meeting the high bar for detecting GABA release from neurons has been actually quite difficult. Mutations that increase the change in fluorescence, see here this gray one, also decrease affinity. And here's the original iGABA-SNFR, something with a higher change in fluorescence, also doesn't traffic very well to the membrane, instead gets sort of stuck in the endoplasmic reticulum. I'll be honest with you that the sensor is not as robust as iGluSnFR. We can see here again doing that experiment where we give an electrical stimulation to a culture of neurons. We got about a 70% increase in fluorescence with iGluSnFR and only about a 10% increase with iGABA-SNFR. You can see that quantified here. Again, the error bars are basically so small you can't see them. Here again, you're only getting about a 10% change, so it's about a 10-fold difference. But it still works good enough in different preparations. So in hippocampal slice, paired pulse electrical stimulation shows a significant increase, about 20% increase in fluorescence, and increasing the extracellular calcium concentration from 2 millimolar to 4 millimolar increases the release probability as expected. Now, in vivo, we can use, this is not we, Dimitri Coleman's group, can use iGABA-SNFR to improve our mechanistic understanding of epileptic seizures. So here we have iGABA-SNFR injected into the visual cortex with a recording electrode nearby. Okay? And this ends up showing the change in fluorescence. Okay? Injection of pilocarpine induces seizures. In the cortex, after seizure has occurred, there's an inter-ictal period that's characterized by these regular electrical spikes with a very distinctive shape. What we see is that the GABA transients basically match up with the electrical activity during these inter-ictal periods with maybe a slight delay in decay. And now, seizures are typified by polyspikes of electrical activity. And during this seizure, we still see GABA release, indicating that the seizures are not caused by a failure of neurons to release GABA, but rather are related to the ineffectiveness of GABA in suppressing activity or some other more complicated mechanism, as we're learning about today. So now, all the experiments I've shown you so far were designed to illustrate the utility of these sensors in detecting basically mostly normal neurotransmission. But there's no reason that we can't use iGluSnFR to query changes in cytosolic concentrations of neurotransmitters, perhaps as a method for assaying GAP1 function. Okay? So that's normal. This is what proposes new. And so this is my last slide from an experiment I did yesterday. So we have, so it's not as pretty as it could be, but here we go. So here we have primary neuronal culture expressing iGluSnFR in the cytosol. So the membrane signal secretion signal has been removed, and the transmembrane domain has been removed. We also have a red fluorescent protein, mRuby3, tagged to the sensor so we can correct for changes in photobleaching, pH, focus, etc. The culture was imaged in the red and green channels, and at 30 seconds, a bolus of high concentration GABA was added. As you can see in the green channel here, fluorescence increased in the green channel, but not the red channel. And when we did a sham addition of just buffer, there was no change in fluorescence at all. Now, interestingly, let's see, which is this. If I draw ROIs around different regions, you'll see that their response is different. If I draw ROIs around what I can tell are glia, we see a pretty high response. If I draw ROIs around individual processes, we get a response, but it's not super high. If I draw an ROI around what I call a mass of processes, which are hard to resolve but are clearly there, we still see a response. And if I draw ROIs around the neuron cell bodies, we do not see an increase, and if anything, there's maybe a slight decrease. So with that, I would like to suggest that this is potentially a good tool for assaying restoration therapies, screening for GAP1 agonists. I'd like to talk with Anna more about that paper that you mentioned. That's going to be an interesting thing to probe. And also to discuss with you, after all this, about what experiments I can do to make an assay more reliable and conductive to doing higher throughput drug screening and probing therapies. Thank you.
Questions?
So you've heard a lot already today about synaptic and extrasynaptic GABA receptors. We'd obviously like to know what the concentration is locally at the synapse versus in the extrasynaptic regions of the neuron. Is there a way of targeting the iGluSnFR?
If you have any ideas, I'm open to those. If you have, so the question was, is there a way to target any of these sensors to the synapse? And the answer is we haven't found one. We have tried multiple different strategies. None of them have been particularly successful. And of course, one of the problems we have is that if we try to hijack some of the architecture at the synapse, we're probably changing the synapse. And now that,
agreed. I mean, Geoffrin is the protein that targets GABA A receptors to the synapse, but if you tie them up, probably the GABA A receptors don't get there.
Yeah,
so
right. The other issue we have is that once we start, if we are targeting an existing architecture, since in our system we are basically overexpressing the sensors, once we have saturated the sensor at the synapse, we then continue to get bleed over into the rest of the membrane. So we have anecdotal evidence that sometimes we can get synaptic localization, but it never lasts very long, and it's remarkably unsatisfying. So open to ideas.
Hi.
Yeah.
Yeah, so first I want to thank you for giving me all the construct. So right now, based on what we had in the construct.
I want to point out, if anyone wants any of these, you just email me. We'll send everything out. And if there's something that you want that we don't have, I'll find a way to make it.
Yeah.
So
right now, based on the mouse models and cell models, we also are going to do microdialysis because this way we can monitor the GABA concentration in brain like, over time.
Mm-hmm.
So what's the advantage of your like iGluSnFR versus like a microdialysis? What condition we use in exercise and neuron versus neuron? So just make some comments.
Right. So the advantages of using the fluorescent sensor are one, time resolution. So we get millisecond time resolution. Maybe that's important. Maybe it's not. We also get localization, down to however fine the resolution of your imaging is. And in terms of prolonged expression, as I said, with some of the variants, we've got an mRuby3 tag on it, so you can probe changes over time. In the iGluSnFR paper, we show that by inhibiting the GABA transaminase, we can see increased concentrations of GABA in the mitochondria, again, over the course of days, normalized for protein expression. So it's frankly, it's really easy. I mean, these assays I did yesterday in like 20, 30 minutes. Well, we can put the sensor everywhere except the synapse. We can put the sensor in the mitochondria with the mitochondrial tag, in the ER with an ER tag.
Only mitochondria is very specific. If it's a mitochondrial promoter, just that. Yeah, I just want to.
Oh, it's not a mitochondrial promoter.
Yeah,
we have it with
specifically localized.
We have a tag. And now, of course, now that you've asked me the name, I can't think of the name of the sequence that we use. I'm spacing on the name. I could look it up on my computer in a few minutes, though. But yeah, there are different retention signals to put them in different places.
Can you multiplex and monitor several analytes at the same time?
Yes. So the slide that I showed with iGluSnFR in the model of epilepsy in the cortex, our collaborators are working with the green iGluSnFR, because that's the only one we have, and one of the other colored variants of iGluSnFR to simultaneously monitor glutamate and GABA at the same time. And then there's also the red calcium sensor, JR Gecko. So we're getting calcium, GABA, glutamate, all at the same time. So that's work that's going on. It's, in theory, completely possible. Technically requires significantly more work.
Hi.
Oh, one more.
Sorry, one more question. You probably have thought of this, but is there a way to excrete it from the cell
Yes,
and then have
very
it
easy.
float and then?
Yes. We've done that. With GABA, it's not a particularly useful tool because it's now just floating around in the extracellular space, and diffusion is fast enough that any signal we get, if we do get a signal, we don't know, we haven't seen one yet, apparently just diffuses out very quickly. We have used this, we also have a glucose sensor, and we've used the glucose sensor just that way to monitor bulk changes in glucose concentration in zebrafish. So, yeah, so it could go. Mm-hmm. But yeah, the problem is that then it's diffusing and you don't really have a good way of capturing it.
It's very cool.
Yeah.
Okay, one more.
And by the way, if there are any sensors that anyone else wants, just tell me, because this is what we do. So if there's another molecule that's interesting, I should know.
So maybe that's my question. So can you tell, so SYNGAP1 protein, postsynaptic, would you be able to look at it?
Sorry,
would you be able to look at other proteins that are haploinsufficiency and what the impact is of the GABA and glutamate, for example, SYNGAP1 haploinsufficiency that's in the postsynaptic gap?
Yeah, so what I would like to do is to be able to use this and with a couple suggestions that people have saying, what is the barrier for, to find out what is the resolution that the sensor can query. So one of the things I'll be doing next week is probing, how do the various GABA uptake inhibitors affect GABA transport as measured by this method. And then once we establish that, that gets back to the question I asked earlier, which was, hey, if we have a panel of 40 different mutants, can we probe the GABA uptake of each of those in a relatively straightforward way? Did that make sense? Okay. Oh, do you have? Okay, thank you.